Hello everyone and welcome to Medical Physiology. My name is John Sullivan and this is the first lecture which I will be teaching fluid homeostasis. Also, let me introduce you to this surly looking gentleman over here. His name is Claude Bernard, and he was quoted as saying, "All the vital mechanisms, varied as they are, have only one object, that of preserving constant the conditions of life in the internal environment." And so that was actually, one of his main concepts was this idea about the internal environment. Okay, the internal environment meaning inside our bodies, right, and how the conditions are maintained. In other words, how we maintain homeostasis. Okay, and there's different things that go on in the body to help maintain that homeostasis, and he refers to those as the vital mechanisms. And that's really what physiology and what I'm going to be teaching you for this semester is all about. It's what are those vital mechanisms? How do we maintain that homeostasis? And so I'm going to start off this semester by talking about fluid homeostasis. All right, what are the vital mechanisms that are going to maintain our fluid homeostasis? One of a multitude of vital mechanisms. All right. Okay, so to start us off, I'm really going to be giving some sort of general information, as you guys will see, some surface level physiology and some concepts. And this is really just to kind of get the gears turning again, to hopefully kind of get some of the rust off, right? And to get us thinking in that sort of mode again, okay? And understand that, of course, I'll be going into more detail as we move along, as we talk about the different organs and systems. I will usually delve deeper into each one of these concepts as we go. So to start us off, very, very simply, all right, we have the levels of structural organization in body systems, right? Very basic stuff. We have the chemical level, carbons, hydrogens, oxygens, and so on, right? These can form the different molecules that are in our bodies, like DNA, for example, and proteins, and so on. Now, this can all, be built together to form in a functional unit known as a cell, right? So that's the cellular level. So all these molecules working together form that cell. Now remember, there's different types of cells in our body. We'll talk about all the different types. The example he gave here is smooth muscle cells, right? But even muscle itself has different cell types. We have smooth, skeletal, right? Cardiac, and we'll talk about all those types in detail. Now, when cells work together, we have what we call it, the tissue level of organization. Now, in tissue, remember, there's 4 types of tissue. Okay, you got the epithelial tissue, which essentially lines the insides of hollow organs and the outsides of organs, and our skin, for example, inside of our mouth. We have connective tissue, which helps to kind of combine and bring everything together, but also serves other functions as well, which we'll talk about. Muscular, and as well as nervous tissue. Those are really our 4 basic types, right? Now, if we take those tissues, those 4 different types of tissues, muscle, nerves, connective tissue, and epithelial tissue, and combine it, we have an organ, right? So the organ they give an example of here is the stomach, right? And you'll see the organs have usually all 4 tissue types. Now, once you get the organ, once organs start working together to carry out specific functions, now we have a system, right? So for example, the stomach doesn't work alone in helping us to digest, right? We also have oral, you have the tongue and the teeth and the salivary glands, and then it's connected to the the intestines, the liver, the pancreas, gallbladder. So there's multiple organs that can work together to carry out particular function like digestion, and we'll learn all about that. And then finally, of course, all the systems working together creates the organism as a whole. So let's talk about the cells for a second here. So I'm going to go quickly to the cellular level. Now, at this level, and in medical physiology, I'm not going to be quizzing you specifically on what these organelles do, but I do suggest that you brush up on some of their functionality. Right? We will be discussing them. You'll probably get it in biochemistry. We definitely talk about it in physiology to some extent, but it's more or less going to be understood that you really kind of know what this is all about. Okay, so if you kind of forget what some of these things do, just remember to kind of brush up on that. But I don't want to spend too much time on it. But what I do want to show you here, and in this cartoon, this is not depicting any particular type of cell. This is a generic cartoon to show you different things that cells may contain. Okay, So for example, you can see the flagellum, right? So let me get my marker over here all set. All right, so here you have the flagellum. Now, again, there's really only one cell type that has a flagellum, and that's sperm. Right? So we'll talk about that, but that's for motility. So it can move through the female reproductive tract. Cilium, right? These are like finger-like processes. These finger-like processes can be found in different cell types in the airways, for example, like the trachea, or you might see them actually in the female reproductive tract or the male reproductive tracts. Over here you can see microvilli. These are all projections off the cell, right? So these microvilli help to increase surface area for absorption. So we might see that, for example, in the digestive tract. If we look, more internally in the cell, of course, you can see the nucleus. Okay, you can see that over there. You can see that connected to the nuclear envelope there, extending out from there. You can see the rough endoplasmic reticulum. It's rough because it has ribosomes, and you remember ribosomes make proteins, or they help to make those proteins, right? Then you have the Golgi complex over here. All right, that's sort of our packaging system, which can then send it to other parts within the same cell, send it outside of the cell, or send it to the membrane to be embedded in the membrane. The smooth endoplasmic reticulum does a lot of detoxifying for us, but also has other functions like storage of, say, high concentrations of calcium, which we'll talk about. And then you see the mitochondria over here. It kind of looks like a little bean. Right, and you will get a lot of information about what the mitochondria functions are, particularly in biochemistry when it talks about its role in metabolism. But of course, we'll touch on some of its features as well. And as you guys know, often called the powerhouse of the cell, produces that ATP. And then there's some others which I haven't quite gotten into right here. But again, we'll come back to this. This is just very basic stuff. But what I really want to focus on the most here in this is the plasma membrane. Okay, so all the cells have a plasma membrane. Now, I will speak in sort of generalities about plasma membranes right now, but understand that there are a multitude of cell types, and not all plasma membranes are created equally. In fact, some are rigid than others, some are more fluid, some have different shapes, as you know. Smooth muscle has like a spindle or a football-like shape, right? Whereas skeletal muscle is more cylindrical, and a neuron has much different shapes even from that. And so that has to do with the cytoskeletal components that help that, but also the membrane itself. Okay, and then the membrane also has things embedded in it. So depending on the cell type, these membranes might have different proteins and carbohydrates and lipids that are in that membrane or embedded in that membrane, and depending on what they are or the arrangement that they have, might give different properties to those cells. Okay, so let's talk about that in a little more detail. So some of the basic functions of the cell membrane are sort of obvious, like a barrier, right? So it keeps the internal environment of the cell separate from the external environment of the cell, all right? And it separates them really by what we call the plasma membrane, which is a lipid bilayer, okay? So that's what I have down here, a lipid bilayer, primarily composed of phospholipids, all right? That's sort of the base substance, if you will. In addition to being a barrier, okay, and separating out external versus internal environments, it actually plays a vital role in controlling the flow of substances into or out of the cell. All right, so this is where the characteristics of individual cells come in. Depending on what proteins and channels and receptors they have, they can dictate how the cell responds to its external environment. What can actually make it into the cell or what can make it out of the cell. And that's where you can have one cell type differ from another cell type is what it will let in or out, for example. Okay, so the example I give here is it can adjust its permeability to various substances. Cells can also, the plasma membrane of the cells can identify that cell. That can be vital when you want to identify the cell as self, meaning it's your cell, belonging to you as the individual. This is important in things like immune reactions to our body, right? So we have something called autoimmune diseases. Auto means self. So self-immune. What we don't want is have a disease where our immune system actually attacks our own body cells. Okay? By doing that, it causes damage and causes a whole host of different types of diseases. Okay? So for the cell being able to identify and tell the immune system, hey, I'm part of you, don't attack me. All right, I'm not a foreign substance, is vital. Signaling. So there will be a whole host of different types of signaling that we'll talk about. especially in endocrine, we talk about all the various hormones, for example, that are released that signal different tissue types. Neurons signal each other, how cardiac myocytes signal each other. There's all varied ways in which it can do that. And it all really depends on how the plasma membrane is built. As I mentioned, the lipid bilayer, which is what the membrane is actually composed of, it's primarily phospholipids. Okay, the phospholipids, you guys usually see that classic drawing where you can see the polar head group that looks like this, and then we see these tails here. Okay, these hydrophobic tails. And so what happens is you have the hydrophilic heads, or the phosphate heads, that face outward towards the external or internal environment where there's more water. Okay, and then the hydrophobic, the water-fearing parts, usually tuck away inside the membrane itself, and the tails face towards each other because they don't want to interact with that water environment. And as you can see here in the electron micrograph of the plasma membrane, this right here is the plasma membrane. You can see one of the organelles inside the cell there. This would be the outside of the cell out here. And you see that bilayer there, the phospholipid heads facing out towards the water. And then you can't see the tails, but these little tails here will be facing towards each other. Inside the membrane is also cholesterol. Cholesterol does play a role, okay? It can increase or decrease the fluidity of the plasma membrane. Glycolipids, right? So this would be sugars that are bound to lipids. And we'll talk about, again, the roles that they play. Proteins, we're actually going to talk a bit about in this lecture. And then, of course, glycosylated proteins, or proteins with sugar moieties, and their roles as well. And so all of these are going to play different functions, or have different functions in signaling and identification and so on. So, plasma membrane, yes, it's flexible, yet it has, somewhat sturdy structure to it, and surrounds and contains the cytoplasm of the cell. Okay, and you can see in the cartoon over here, you can see those phospholipid heads that I talked about, all right, and tails facing inward away from the water, which would be here inside the cell, which is the cytosol, and then the extracellular component, which would be in the white area there. And this is just a cartoon just to kind of depict or show you how you can have different things embedded inside the membrane itself. So for example, these are little cholesterol components of the lipid membrane there. And you can see things like proteins. Now proteins can span the entire length of the membrane, so that it's facing, one side's facing the external environment, one's facing the internal environment of the cell. You can have some proteins that only stay on the periphery, meaning only on the outside surface of the cell, or on the inside surface of the cell, or only partially go in, and so on. So there's different arrangements that they can have. You can see here in purple some of the carbohydrate moieties that could be bound to lipids or to proteins. Now, in terms of it acting as a barrier, okay, which is one of its major functions, is to say, what can move through that membrane. There's really two ways that it's kind of selects, and it's either an energy dependence, meaning it requires ATP, or it's a passive process. So let's start with the passive. If it's passive, meaning it doesn't require any energy, in other words, it doesn't require any ATP. So we have really two main types, which are diffusion and facilitated diffusion. With diffusion, really what this means is we go from high concentration to low concentration. That's the natural pattern for molecules or solutes that are dissolved in solution to move. They want to go from high to low, right? And the reason, again, this is really just sort of random movements, okay, of the molecules when they're a lot of them next to each other. They're bouncing off each other and they start to separate more and more. And that's really it diffusing away. And when it diffuses away, it goes, again, down its gradient. And so that's the natural way most molecules want to go, is down their gradient, okay? Now, in terms of simple diffusion, if you have a very small molecule, or it's soluble in lipids, meaning it could move through that lipid membrane, right? It could diffuse very easily. So you could have a high concentration outside the cell, and it would diffuse down its gradient into the cell, okay? And it could diffuse passively, not requires any energy. It can move right through the membrane. In fact, anywhere along the membrane, it can move in. It can move down its gradient into the cell, and that's referred to as simple diffusion. Okay. Restricted diffusion is like simple diffusion, same concept. It requires a concentration gradient. Okay, but in restricted, it additionally requires the fact that it needs proteins or channels to allow it through. So it's restricted because it needs to go through particular channels. All right. So for example, this would be charged molecules, okay, ions. Say, take sodium, for example. Sodium is Na+, all right? So this little charge here makes it difficult for it to move through a membrane, okay? Because the membrane is lipid-based, and so it's not going to allow things like charged molecules or polar molecules to move through a lipid membrane easily. So it can't just simply diffuse through the membrane. It's restricted. It has to go through specific channels. And as you will see, some channels are constitutively open, some require signaling, and so on, and we'll talk about that. But again, it's a passive process. Facilitated diffusion, again, a passive process. The difference here would be a molecule that wants to go down its concentration gradient, only now it needs to bind to a specific receptor on the cell that's, that recognizes its shape. Once it recognizes its shape, it can move it down its concentration gradient into the cell. A classic example of this would be glucose transporters, right? It specifically will bind to the glucose molecule because of its shape. And it'll move it down its concentration gradient once it recognizes the shape and moves it in, okay? Active requires, ATP, requires the use of energy. When we would usually use energy, all right, is when we want to go against the gradient, when we want to go from low to high. That's not the general way of things. We don't want to go from low to high, we want to go from high to low. So if I want to move something against its gradient, say out of the cell, from inside the cell to out of the cell against its gradient, then I would need to utilize energy, okay? I'll show you some examples of that, and we would refer to this as primary. So for example, if the transporter that is moving something against its gradient directly hydrolyzes ATP, for example, all right, then it's considered a primary active transport, meaning that transporter is directly using ATP. Now, once it sets this gradient, okay, it's moving molecules against their gradient to increase concentrations, let's say, outside the cell or inside the cell, there might be other receptors, okay, or transporters in the membrane that want to utilize that gradient because there's a lot of potential energy in a very high gradient, okay? So in order for it to do that, it needs the primary to set that gradient, and it takes advantage of that gradient and uses that gradient to help drag other molecules against their gradient. It's a little bit confusing, I know, but I'll describe this in a little bit more detail soon to come with some examples. But essentially, secondary requires the use of primary first. So primary has to set the gradient, secondary takes advantage of the gradient to help move other molecules. Proteins. So when it comes to the proteins, all right, they are quite varied, as I've kind of already mentioned. So I want to talk about some of the different types that we're going to run into. All right, you have the channel type. So it opens up a pore, and that extends the distance of the membrane from the outside of the cell to the inside of the cell. So we usually refer to that as integral. So it's an integral protein. And you can see the pore over here. You can see some of the molecules moving through that pore or that channel. And this is often what you're going to see with these, what we call ion channels, right? So ones that are, maybe specific to, say, sodium, potassium, calcium, chloride, and so on, okay? Now, some of these channels can also be voltage gated. And we're going to talk about this in more detail in a specific lecture, but this basically is to say that, the cells in our body, inside the cells can have certain voltages, right? Usually what we call millivolts. And if that voltage changes due to some sort of stimulus, if it changes, depending on what that new voltage is, that might trigger some of these channels to then open or close. So they are basically gated by the changes in voltage. They may have what they call a threshold. And if the voltage changes and reaches that threshold, then that channel may open and allow ions to move down their gradients, or it may close, depending on the type of cell we're talking about, or the type of voltage gate. There's also ligand gated. Now, a ligand is really any molecule that can interact with one of these proteins. Typically things like hormones, for example, or something like acetylcholine, right? So the example I give is acetylcholine. Acetylcholine can bind to one of these channels, and it can open up what we call a cation channel, and it'll allow things like sodium, potassium, calcium to move through the channel. But it requires the binding of acetylcholine. So if acetylcholine isn't present, then it stays closed. All right. So this is just showing you that there's sort of a multitude of ways to activate these channel types. And as we'll also see, some of these channels may actually just be constitutively open. Okay, so there's other types of proteins like transporters or carriers. They may require energy, or they could be passive. Right? So it could be primary or secondary active transport, or passive, like in facilitated diffusion. So an example of a facilitated diffusion type would be here, which you can see, that's the glucose transporter. And so it's, let's say after a meal, the glucose concentration has increased in our blood, and we want to get that into the cells so that we can, break that down for energy requirements. And you see here's the glucose transporter. It has a specific receptor there that will bind to glucose and transport down its concentration gradient inside the cell so we can metabolize it. Okay, then that would be an example of sort of a passive way in which we can move glucose down its concentration gradient. Okay, another type would be over here, which you can see is the often referred to as the sodium-potassium pump. This is really an exchanger. What this is doing is this sodium-potassium pump is going to move 3 sodiums out, so 3 sodiums out and 2 potassiums in. All right, and so it's moving sodium out against its gradient. Remember, which we will talk about. Actually, I shouldn't say remember because we haven't talked about this yet. But as you will see, sodium is in high concentrations outside the cell more so than inside the cell. And potassium is in high concentrations inside the cell more so than outside the cell. And this is going to be something we're going to see a lot of, so it's good to kind of remember it now, which is why I'm reviewing it with you right now. So these pumps help to maintain this gradient. Okay, high sodium outside the cell and high potassium inside the cell, because it's switching them, and it requires energy directly. So you'll notice here, it's utilizing ATP, the ATP which we probably got from the mitochondria there. All right, it directly hydrolyzes it to ADP, and you'll see that we now have the energy required from that hydrolysis reaction to pump sodium against its gradient and move potassium inside the cell against its gradient. Now, in terms of secondary active transport, oftentimes what you're going to see, and I'll just sketch it over here. Let's say I have, we'll draw over here, I'll draw another one over here. Okay, and so let's say this one right here is our sodium potassium pump. All right, so it's pumping out sodium and it's pumping in potassium into the cell. So let's say this is inside the cell and this is outside the cell, and this is our plasma membrane right here, so plasma membrane. Okay, and then over here, let's call this one a sodium calcium exchanger. So just like our sodium potassium pump is an exchanger, it's exchanging sodium with potassium. Here we're going to have now a high concentration of sodium outside the cell thanks to the actions of our pump here utilizing ATP. So it's primary active transport. The sodium calcium exchanger doesn't directly utilize ATP. Instead, what it does is it uses that very high concentration of sodium and says, all right, well, I'm going to let sodium go down its concentration gradient. So I'm going to let sodium go down its concentration gradient inside the cell. But in return, what I'm going to do is I'm going to pump out calcium, 2+. I'm going to pump out calcium against its gradient. So this is calcium. Calcium is another ion that primarily resides outside the cell, not inside the cytosol. So this is one way to get calcium out against its gradient. It didn't directly utilize ATP, but it requires the actions of the pump right here. If that pump didn't set the gradient, then there would be no gradient for sodium to move down its concentration gradient to pump calcium out. So the sodium potassium pump doesn't work, the sodium calcium exchanger can't work. So it relies on it. So that's what a secondary active transport does. It relies on that primary pump. There's a multitude of additional types of proteins with varied functions, but just to kind of touch on what some of those are, there are receptors, and you can see over here to the right the little cartoon that I have here for you guys. Receptors are integral proteins. They may not necessarily be channel. We've talked about receptor channels, where you could have something like acetylcholine bind, open up a channel, and allow ions to pass through. Okay, those are very quick acting. However, you might get receptors that when bound to a ligand, maybe acetylcholine, could bind to it. Instead of opening up a channel, it just causes a host of signaling to occur inside the cell. So something inside the cell may be bound and be responsive to that receptor. So when the ligand comes along and binds to it, it activates a cascade of events that takes place internally in the cell. Enzymes. You'll see that many different types of cells are going to have enzymes embedded in the membranes, and as you guys know, enzymes help to catalyze chemical reactions. So in one of the examples here, which we have, one of the enzymes is integral, and this one, in this example here, is actually facing outward outside the cell. And this is actually a classic example of an enzyme that faces outward in the intestinal lumen. So remember, the intestines help with digestion. So in this example, if you have lactose, okay, so lactose will bind to this enzyme on the surface of these cells, and that enzyme will cleave it into its products, right, glucose and galactose, so that it can be absorbed by the cells. So that's an enzymatic reaction that's occurring right on the surface of the cell by an enzyme that's embedded in the membrane of some of those cells. Which, of course, if somebody's lactose intolerant, they may not actually have those enzymes to be able to carry that out. Linkers. Linker proteins, well, they're, as their function suggests, they link cells together. Okay? So they could actually play a vital role in keeping cells adhered to one another, particularly in, like, say, epithelium, like your skin, for example. These cells have to keep bound to each other, otherwise they would fall off and fall apart. Okay, so it helps to keep cells together. It also links cytoskeletal components within the cell itself. Okay, it helps to kind of anchor them. It helps form their shape. Glycoproteins. Glycoproteins, these are sugars bound to the protein. So one of the classic roles for them would be something like cell identity or a marker, like a major histocompatibility protein, which you see here is MHC. This is, as I mentioned earlier, is so that the cell can mark itself as being part of you, self, right? So it's a self-identifier and helps to let our immune system know that it's not foreign. Okay, so one thing I like to do is, with all the basic science that you guys are learning, is to kind of step back and look at it from a clinical perspective, so you guys can see how this, what you're learning, how it pertains to what you guys want to do. So, for example, loss of anchoring membrane proteins. There's a disease called Pemphigus vulgaris. Okay, this is an autoimmune disease that actually attacks cell junction proteins or linker proteins, specifically ones known as desmosomes. Now, as you recall, those linker proteins, which I just spoke about, they help to keep cells connected to one another. And in the skin, for example, that can be particularly important to make sure that the skin stays attached and cohesive and it stays anchored to your body. And so if your immune system is attacking those proteins, okay, and you lose the ability to actually adhere cells together, you could actually lose your epidermal layer of your skin. And clinically, you might see signs like this. Something called Nikolsky sign. Now, this is not specific at all to Pemphigus vulgaris. There's other conditions that can cause this. But what happens is, you can actually gently rub your finger across the surface of the patient's skin, and they will easily exfoliate the layer of their epidermis. You can see here it just kind of peels right away. Okay. Oftentimes you see lots of blistering on their skin. The skin just kind of can exfoliate right off. Okay. And so what we look at under the microscope, which you can see down here, all right, just to kind of orient you guys here, this entire layer from up here to here, this entire layer is part of the epidermis. Okay, down here, which is, you can see all this pink stuff down here, that is the dermis. Dermis. Okay, this is the epidermis on top of that. That's the outermost layer of your skin there. And you'll notice this little whitish area here, sliced open there. So basically what that looks like is, since you lose those linker proteins, those cells that are supposed to be able to adhere to each other there and hold on to one another, they're being attacked by the immune system, and so there's no longer the ability to adhere there, and the skin can slough off. And you might ask, what's the danger of that? Well, besides the fact that it can look fairly scary like that, but it can actually present like burn. Our skin is an effective barrier to protect us from things like infections, so we're at higher risk of something like an infection, and also dehydration, because the skin helps us to protect and conserve water. So we might lose more water than we should. All right, so now let's talk a little bit about those receptors. Now I talked about receptor channels, which are, again, those ligands bind and open up a channel. But now you have the other type of receptors. These type of receptors don't open up a channel. Instead, it causes a cascade of signaling that occurs inside the cell. Oftentimes, it's referred to as a metabotropic receptor, versus an ionotropic receptor, which is a receptor that opens up a channel. So how does a signaling molecule activate cellular activity, right? So we have our ligand, and this is one example, by the way. There's a multitude of different examples here, but here's one. We have our ligand. Again, any particular hormone, we can pick one. Let's call it epinephrine. Okay. So epinephrine, or sometimes referred to as adrenaline. Okay. So we have our epinephrine binds to the receptor at its specific site right here, and here's what we call a GPCR, or a G protein coupled receptor. So again, I have it over here, G protein coupled receptor. Okay. Now on the inside of the cell, that G protein coupled receptor is bound to this structure right here. All right, these proteins, which have several subunits, and alpha, beta, and the delta subunits right here, also bound to something called the GDP. All right. When the ligand binds, that GDP switches to a GTP. Is replaced by a GTP. Okay. You'll notice that it separates from the other subunits. So the alpha subunit bound to the GTP then can interact via a protein that's linked to that G protein coupled receptor, which is called adenylyl cyclase. Adenylyl cyclase is an enzyme. So I spoke about enzymes. The enzyme I gave was an example of one that faces outward and can help with digestion. This enzyme, on the other hand, faces inwards and actually converts ATP into cyclic AMP. Cyclic AMP is one of the most common second messengers that you're going to see. In fact, I've listed over here second messengers. Number one, cyclic AMP. Another one might be cyclic GMP. So instead of adenylyl cyclase, you'd have guanylyl cyclase. It's a different type of G protein coupled receptors that can do that. Instead, the G protein coupled receptor might activate a pathway that activates IP3 and diacylglycerol, or DAG. That increases intracellular calcium, for example, in cells that are calcium dependent or calcium requiring activities like muscle. 4, arachidonic acid. Okay, that actually helps in inflammatory responses and hemostasis, which we'll talk about. So this is just an example where you have the cyclic AMP, second messenger, can then move inside the cytosol and activate pKa, or protein kinase A. And just to remind you guys, a kinase is a protein that phosphorylates something else. A phosphatase removes the phosphate group. And remember, the phosphate can turn on or turn off proteins and so on. So it depends. So usually what we say is it activates the pKa, and then depending on the cell type, we'll get certain responses from that. Okay, it can be quite varied. So for example, if it's epinephrine and this was a cardiac myocyte, when the epinephrine binds to the G protein coupled receptor, that's going to increase cyclic AMP. All right, and then that cyclic AMP increases or activates the pKa's, and the pKa's in turn will cause the cellular response in the cardiac myocytes, which would be to increase heart rate, increase its contractile strength, for example. Now, important to point out is that even though epinephrine is doing that to the G protein coupled receptor in the cardiac myocyte, the G protein coupled receptor that binds to epinephrine in a hepatocyte or a liver cell is an entirely different function. So if epinephrine binds to, say, the G protein coupled receptor on a hepatocyte, that's going to carry out something like glycogenolysis, where it breaks down glycogen to release glucose into the blood. So it was epinephrine, it was a G protein coupled receptor, but depending on the cell type, it was totally different functions. So understand though, this is where we get the diversity. Despite the fact that we have the same hormones or even the same receptors, depending on the cell types, we can get a host of different responses from the body depending on what their cells were that were responding to it. Other major types of cell receptors, tyrosine kinase dependent receptors. We'll touch on that a little bit in the metabolic lecture, but that's basically, one of the classic examples is insulin. Insulin binds to tyrosine kinase receptors. They dimerize and autophosphorylate and so on, and that causes glucose receptors to go to the surface of the membrane to increase their uptake of glucose. Receptor channels, we talked about that already. Those are the ionotropic receptors, but you can also have intracellular receptors. So not all receptors are located in the plasma membrane. Some of the receptors can be located in the nucleus or in the cytosol. And so if you have, say, a lipid-soluble hormone, like a steroid, for example, like cortisol, that can actually travel through the membrane, plasma membrane, and interact with receptors inside the cell. But we'll talk more about that when we get there. Okay, so in broad stroke terms, we have our homeostasis. I'm coming back to homeostasis. So that was our overview of cell membranes and proteins and how it acts as a selective barrier. In terms of homeostasis, going back to Claude Bernard from the beginning, we're talking about the vital mechanism. So now we're finally getting to the point of this lecture, which is fluid homeostasis. Okay, so before I begin though, just keep in mind, like, what are the different vital mechanisms that we use? Like, what are the different things that we try to maintain homeostasis with? Right, so can you think of any kind of normal ranges? Right, so for example, glucose. Glucose has to be kept at a certain concentration in our blood. CO2, oxygen, right? The pH of our blood pressure, right? So these are all things that have to be regulated and have to be maintained within a range. Doesn't have to be maintained exactly. So for example, if glucose is 90 milligrams per deciliter, is a sort of our normal, okay? It doesn't exactly have to be 90 milligrams per deciliter, it's within a range. Within that range, we'll still have normal operation, okay? Some ranges are smaller or tighter than others, as you guys will see, all right? But this is about the mechanisms that are going to maintain us within those ranges. So the first one up is fluid. Okay, so in terms of body fluid homeostasis, understand that the survival of our body cells is actually really dependent on precise regulation of the volume and the chemical composition, all right, of the surrounding fluid that's outside and inside the cells. Okay, so this is vital. So a lot of actually our energy, of our basal metabolic rate, a large percentage of that energy is actually directed towards maintaining this precise regulation, making sure we have the precise concentrations that we need, all right, and the volumes are maintained accordingly. So in terms of the breakdown of those volumes, okay, what we have over here, you can see in this little pie chart, the intracellular fluid, okay, which makes up the majority of the fluid in our bodies. And then the extracellular fluid. So just to be clear, all right, fluid inside the cells is intracellular, okay? The extracellular fluid, otherwise known as the ECF, that's the fluid that fills the spaces around the cells, as well as in the intravascular space or in our blood vessels. Okay, so the fluid that's around our cells is known as interstitial fluid. Okay, and then the fluid that's actually in the blood vessels is referred to as plasma. And you'll notice that the extracellular fluid is primarily interstitial fluid and plasma. And then there's a little sliver here of other fluid, which is highlighted in green there. That other fluid really pertains to down here, which is what we call transcellular fluid. Like, for example, pericardial fluid, like the fluid that surrounds the heart inside the pericardial sac. Cerebral spinal fluid, the fluid that surrounds our brain and spinal cord. Synovial fluid in our synovial joints. So there's, pleural fluid that surrounds our lungs. So this would be considered transcellular, but in in normal conditions, this is actually a very small amount of fluid. But ultimately, this could become pathologic and could be a very large amount of fluid in certain conditions, like tamponade, which is large amounts of fluid in the pericardial space that have accumulated there that could compress the heart, for example. What you're seeing down here is sort of a classic picture of edematous feet, where there's an imbalance of water regulation. You see edema. In fact, you can see little fingerprints from where they pressed on the edema, and that's referred to as pitting edema, something you guys will learn more about as we go along. So let's take an example of sort of our daily intake and loss of water. Understand that since we're in balance or in an equilibrium, whatever we take in, all right, we have to also void. So we have to get rid of, right? So if, for example, we have a 70 kilogram person, and on average their daily day, okay, from resting to, to exercise or whatever, let's say their fluid intake is 2,300 milliliters of water. All right, that's 2.3 liters. So if that's their intake throughout the day, right? Urine output is typically only about anywhere from about 1 liter to 1.5 liters, or 1,000 to 1,500 milliliters if you want. Okay. So how do we get rid of the rest of the fluid? Well, the rest is lost in feces, okay, sweat, something called insensible loss. Insensible loss is different from sweat. All right, this is actually evaporation through the skin and lungs, primarily. Okay, this is separate from sweating itself. Okay, in fact, I've seen studies where they've had genetically modified animals who have lost their sweat glands, who still lose water through evaporation, called, this insensible loss. So let's take a look at an example here. So here's our normal condition. So our intake, so we have fluids that have been ingested. So this person has ingested about 2.1 liters, all right? And then from metabolism, all right? Remember when we catabolize, for example, glucose, right? Water is produced. So yeah, when we break down some of those foods, we can also get that. So from metabolism, we get another 200 mLs. So that adds up to a total of 2.3 milliliters, or 2.3 liters of water. Let's see what our output was that day. So insensible skin, all right, that means evaporation through the skin, about 350 mils is average for a 70 kilogram person. Insensible lungs, 350. That's 700 mils. That's almost 1 liter. Okay, so 1 liter of water that has been lost just through evaporation. Sweat, all right, so sweat about 100. You see feces, another 100. Urine, our typical, again, between 1 and 1.5 liters. It's about 1.4 liters here. That total is going to be about 2,300, and you'll see that that's an equilibrium of intake and output. Now, an example, if we're, say, doing heavy exercise. Well, let's just say, for example, we don't know what they're ingesting during heavy exercise, okay? Metabolism, let's say it's about 200 still, but we don't know what the total intake is right now. However, as far as the fluids lost, this is just to kind of point out something here. You'll notice that the evaporation through the skin stayed about the same, but insensible loss through the lungs, all right, just about doubled. Again, they're also respiring more, they're breathing faster, so we get a little more evaporation. Body's also heating up, so we got a little more evaporation through the lungs. But look what happens to sweat. Sweat went from 100 to 5,000, so there's a lot more sweating going on, all right? Feces stays about the same, and urine output goes down. Urine output would go down during exercise as we try to conserve some of that water. So that's an appropriate response by the kidneys to reduce urine output. Okay, and we'll talk more about the specifics of why that is later on. But look, during heavy exercise, you'll notice 6.6 liters. Okay, so that means to match that, if it was heavy exercise, they would have to drink, 6.4 liters to be in equilibrium. Okay, but this is just to kind of show you how these fluids are lost. Places that we don't tend to think of it. We tend to think of it as, input, right, as we drink the water, and we tend to think of it as output through the urine. But there's actually a multitude of ways, and depending on our state, whether we're exercising or resting, those can change pretty drastically. Okay, now in terms of calculating total body water, all right, there's a few conversions that are helpful to keep in mind. Understand that in a clinical scenario, oftentimes you'll get the patient's weight in, say, pounds, okay? But most of the calculations are easier, all right? They're easier if we convert them into, kilograms. All right, you're going to see that in pharmacology. It's going to be easier for calculations like total body water and so on. So it's something that you got to keep in mind. And there's a couple of useful tips that I put in here for you. So for example, before I begin talking about water percentages and whatnot, conversions. 1 ml of pure water is equal to 1 gram of water. So that's the mass or the weight of the water, right? So if you have 1 ml of water, that's about 1 gram of water. So again, if I were to convert that, instead of a milliliter, if we're talking about in liters, I have to multiply it by 1,000. So 1 liter of water is equal to 1 kilogram of water. All right. So these, again, this conversion is useful to keep in the back of your mind. Also, calculating total body weight in kilograms. Since most of these calculations are done using kilograms, this is useful to know. So again, if you get the weight in kilograms and you want to know it in pounds, well, 1 kilogram is equal to 2.2 pounds approximately. All right. So for example, if you have a 70 kilogram man, all right, you want to know what that is in pounds, multiply it by 2.2. That's 154. All right, 154 pounds. On the other hand, if you get it in pounds and you want to convert to kilograms, you divide it by 2.2. Okay, so you have a 190-pound individual, divide that by 2.2, you get it in kilograms. All right, so that's something to keep in the back of your mind as we move forward. Now, in terms of body percentages, so here what you can see in the cartoon, males and females do differ from each other slightly. So females, generally the total body water or total body fluid is 50 to 55% of their body composition is in the female adult. In the male adult, it's 60%. Okay, where the rest of it is going to be, solid and so on. So let's take the male for example. I'm just going to use the male in the sample because 60% is a little bit easier to work with than 55. But here we have 60%. 2/3 of that total body water, 2/3 of it is going to be intracellular fluid. Okay, so 2/3 of that. Now 1/3 of that is going to be extracellular fluid. Now remember, if you recall, I talked about in the previous slide, extracellular fluid is primarily the interstitial as well as the plasma from the blood. All right, so 1/3 of that is divided amongst the interstitial and the plasma. So that's why you're seeing over here the extracellular fluid, 80% approximately, right? 75 to 80% is going to be interstitial fluid, and 20 to 25% is going to be the blood or plasma. Okay, and so that's going to make up the extracellular fluid primarily. All right. Now, in general, females tend to have less total body water because they have a higher adipose tissue. Remember, adipose tissue is fat tissue. Fat tissue, remember, fat repels water for the most part. So if you have adipose tissue, it doesn't hold on to a lot of water. And so the more fat content somebody has, the lower their water content is. Now, lean muscle, on the other hand, or lean tissue, like skeletal muscle, for example, contains a lot of water. So the more lean mass that somebody has, the more water that they have. So their body water content could be higher. All right. Now, it's important to understand I'm talking about adults right now, but infants, they can actually be 75 to 80% water, which is interesting. So there's 75 to 80% water. As they age, right, by about 1 to 2 years old or so on, that goes to the normal percentages of like 50 and 60% for males and females. But as we get older, become elderly, the elderly, they actually lose a lot of lean muscle, which contains a lot of water. They lose all that, a lot of lean muscle, and they gain more adipose tissue, which has less water. So you'll see the body water content, or the total body water, that level goes down. Okay, that total body water goes down. So that, and let's say a male would be 60%, he might be at 45 or 50%, in his 70s or 80s. Okay, so I want to work through an example with you right now using that breakdown in total body water. So I'm going to use the example I kind of gave already, which is, let's say we have a 70 kilogram male. Okay. Now 70 kilograms. Now remember, with 70 kilograms, I could have given that to you in pounds. So I could have said, I have a 154-pound male, right? So 154-pound male, divide that by 2.2, and that would have given me the 70 kilograms. Now, if you recall that if I want to know what the total body water is, total body water, that's equal to 60% of his body weight in kilograms. Right? So basically, I just multiply 0.6 times 70. Right? So I can say 0.6 times 70, and that's going to give me 42 kilograms. All right, since that's 60% of 70 is 42. Now that 42 kilograms, if you recall from the previous slide, I was telling you about the conversions, and I told you that 1 kilogram of water is equal to 1 liter of water. Okay, so I can actually convert. So if I know that 42 kilograms, all right, which is 60% of his body weight, is actually going to be fluid, I can convert that. So 42 kilograms is actually 42 liters. So this 70 kilogram male on average would have 42 liters of water. So that's his total body water right there, 42 liters. Now, the next thing I want to know is how much of that is intracellular versus how much is extracellular. So let's take a look. If I have my intracellular, or the ICF, and my extracellular fluid, or my ECF, and I want to know how much is intracellular, remember that 42 times, now what percent is intracellular? So that's actually going to be 66%, or 0.66, or in other words, 2/3, as I mentioned before. Okay, so that's going to equal 28 liters. All right, and now I don't have to do the a calculation here. I can just subtract 42 from 28, but I'll know that that's going to be 14 liters. Will be the final third there. Okay, so total intracellular fluid is 28 liters. Total extracellular fluid is 14 liters. Now the last bit I want to know is, say, let's say I have the extracellular fluid. Remember that breaks down primarily into interstitial as well as plasma, blood plasma. What's the intravascular? All right, so I want to know interstitial versus plasma, how much fluid is in each one of those compartments on average. So if you recall, interstitial primarily has about, say, 80%. All right, so I'll put that here as 80%, and this is typically 20%. All right, now 80% of 14 is going to be about 11 liters, and 20%, then of course I could just subtract it, would be 3 liters. So now I have a breakdown of basically the amount of fluid that he has. So the total amount of fluid, which would be 42 liters, 28 of which is going to be inside his cells, 14 is going to be in his extracellular fluid, the fluid that surrounds the cells, and is also in the blood vessels. And of that 14, 11 is going to be in the interstitial space, and 3 of it's going to be in the blood vessels as plasma. Understand that I specifically, and I'll put a little asterisk here, say plasma and not blood. Because when we say blood, we're really referring to whole blood. Whole blood also contains cellular components, like red blood cells and white blood cells, right? As well as things like platelets. When we add the platelets, the red blood cells, and the white blood cells to the plasma, which is really the fluid portion of the blood, then the volume would go up to, say, about 5 liters. And in fact, your average male, about 70 kilograms, typically has anywhere from 4 to 5 liters of blood. However, I'm calculating what is the fluid portion of the blood, not the cellular component. Right? I want to know about the fluids. So when we talk about plasma, we're talking about the fluids. That is 3 liters. Okay? So that's 3 liters of blood. Now, there is a quick calculation that you guys can utilize when it comes to doing this, which is, say I told you I had a 70 kilogram man, okay, or any weight really. You can do what we call the 60-40-20 rule. And what that states is that 60% you'll get the total body water. So multiply his weight by 60% and you'll get total body water, which in this case was 42 liters. If you multiply his body weight by 40%, you'll actually get the intracellular fluid volume, because 2/3 of 60% is 40%, right? So 40% of his body weight, that'll actually give you 28 liters. And lastly, if you just multiply by the 20%, which is the final third, that'll give you the ECF, which would give you 14 liters. So the 60-40-20 rule works best, of course, for males, but for females, really there's about an 8% difference or so. So you could decrease that by about 8%. So you could work with, more like a 50% and go from there. But this is really more common. You could even use this for females too, particularly if they're fairly lean. But yeah, the 60-40-20 just lets you know that you can use this breakdown. Okay. Of course, if you wanted to know interstitial versus plasma, you'd still have to further break down the ECF and remember that 80% is interstitial and 20% is in the plasma. So that's just some basic ways to calculate the total body water. And then really what's very useful is to be able to break down to the ECF and figure out how much is in the plasma, for example. So a couple of things, the body maintains homeostasis of the interstitial fluids and the plasma via thirst mechanisms. So the hypothalamus lets us know, when we're thirsty and when we need to drink. And then, of course, we have, if the kidneys are working, appropriately, we'll excrete the appropriate amount of sodium and water so that we stay in that balance, okay? Now, this cartoon over here, which you can see is the cells, and this is again just to help you kind of visualize interstitial and plasma versus intracellular. You can see the two cells here. We have the ICF, the intracellular, and that's going to have that 2/3 of the total body water, okay, which we've discussed. And then surrounding the cells, we have the interstitial fluid. So that's all this sort of like tan color over here, the ECF. And the ECF also includes, again, inside the intravascular space or inside the blood vessels, the plasma, right, the fluid portion of the blood. Again, 80% in the interstitial, 20% inside the plasma there. That's going to be together, that's going to combine to make about 1/3 of your total body water. So I give you a quick tip down there again, just as a reminder. Now, again, we can't talk about just body water without talking about what's dissolved in it, right? So what's actually inside the water, because for us, water is our solvent, okay? But we have lots of solutes that are dissolved in that water, albeit most of them are in very, very tiny concentrations. Okay, so oftentimes when we're talking about the concentrations of solutes inside dissolved in the water in our bodies, we're talking about milli amounts, like millimoles. Okay, so these are very tiny amounts, or even micromoles in some cases. Now, just to give you an example here in this cartoon, you see the extracellular fluid, okay, and there's the intracellular fluid. And so let's take a look at some electrolytes like sodium, potassium, here's calcium, magnesium chloride, and so on. And here's the concentration. And again, the concentration is, either the weight or the amount of a substance dissolved in a volume. All right, so here's giving it to you in what we call milliequivalents. So sodium, you have 142 milliequivalents per liter. You'll notice that inside the cell is only 10 milliequivalents per liter. And physiologically, actually, our cells maintain that. They maintain low sodium inside the cells, high sodium concentrations outside the cell. The reverse is true with potassium. You notice that potassium is 140 milliequivalents compared to 4 milliequivalents. So it's reversed. In fact, I've already talked about this or alluded to this in the earlier slides when I talked about the sodium-potassium pump. How sodium is moved out against its gradient, right, out of the cell. So the cell is able to maintain that using active transport. And this just gives you an idea of how some of the other ions compare. So like calcium is primarily located outside the cell compared to inside, right? Then magnesium, chloride, bicarb, phosphate, and so on. And so you can kind of compare, the different concentrations from inside relative to the outside. Now, you'll notice here at the bottom, urea, all right, 15 milligrams per deciliter versus urea 15 milligrams per deciliter inside the cell. You notice it's about the same. And the reason for that is urea is a small lipid-soluble substance that readily moves across the membrane. So it can easily equilibrate from the outside to the inside of a cell. All right. So the other thing I wanted to kind of point out, which you probably may already have recognized yourself, is that the units that are being used. Most of the electrolytes we looked at have been using units like milliequivalents per liter. And it is okay right now if you're saying to yourself, I don't remember what a milliequivalent is or what that's, what that represents, and that's okay. All right. I'm going to go through that in the next few slides, but understand as I go through this, I'm not expecting you to do chemistry type calculations. All right, but I go through it in a little more detail simply because I want you to have a good understanding. So when you see these units, you're not confused by them at all right? Because you're going to see them quite often. And not all labs are going to give you the same units. So here, again, we're talking about milliequivalents. So I'll come back to what that is in a few slides, about what a milliequivalent per liter is. And I also use, say glucose milligrams per deciliter. So that's looking at it from, mass per volume. So we'll talk about that in a few slides. This is actually an example of something you might see, right? A blood chemistry panel, all right? Sometimes referred to as a complete metabolic panel. And this is actually, from a patient. And you can see here, sodium, potassium chloride. We look at all these electrolytes because they are important. These are important solutes in maintaining our homeostasis. And so the levels have to be maintained, right? In order to carry out our vital functions, right? To maintain our homeostasis. I mean, yes, they will give you reference ranges on most of the lab sheets over here. And they will indicate to you if any of them seem to be high or low with the, they'll bold them and put an H or an L there. But understand that we're going to learn how to interpret that, okay? It's not enough to see something is high or low or out of range, all right? It's important to understand what that means, okay? Particularly in the context of the patient. So since I don't know anything else about this patient, I can only give you whether it's high or low. I can't really say much else about it, okay? And as far as knowing what the normals are, if you don't know them now, by the end of my physiology class, you'll be very familiar with them. Okay, that much I can promise you. You may not remember all the ranges, but you'll be able to recognize whether something's normal or not. Okay, so say sodium, all right, gives you the reference range, 135 to 144, and you'll notice the units here are millimoles. Before, we just looked at milliequivalents. Now they're using millimoles. I think in the previous slide it was about 144 as well. So what does it mean? What is milliequivalents per liter versus millimoles per liter? We'll talk about that just in the next slide. All right, but take a look, 144 and 4. What this is this is a blood chemistry. So this is only going to be representative of the concentrations relative to what's in the blood, okay? Because we're not taking it from inside the cell, right? So here you can see like chloride's about 109, CO2 it's taken, and anion gap, which we'll talk about later on. Something called osmolality, which was calculated. You're going to learn how to do an osmolality as well. All right, and we'll learn what that means for us, and as far as the patients go. All right. Now I look down here. The units they're using are in some cases milligrams per deciliter, grams per deciliter. So in grams, this is much. This is 1,000 times more than the milligrams. So things like proteins in your blood, like albumin, globulins, and so on, they use units of grams. So that means it's quite abundant, especially relative to, say, something like calcium, which is milligrams. Okay, so here's where we get to the fun part of the lecture, where we talk about units of concentration. So I don't expect you guys to have to do like a ton of these conversions in chemistry style. What I do really look for is a basic understanding, maybe able to do simple and quick calculations with some things that you guys will see quite frequently. All right, so if you can kind of arm yourselves with that, you guys are going to be ahead of the game. So let's just start. Concentration is an amount over volume. Simple enough. Now we can look at concentration as a weight per volume, where we would use units like grams or milligrams per a given volume, like liters, milliliters, or deciliters, right? So the example I give here is a weight per volume, say grams per liter, and I bolded milligrams per deciliter because we've actually seen that already. I talked about that on the previous slides where you saw something like calcium even was given as milligrams per deciliter, or urea or glucose. All right, so that's a very common units to see. And it's a weight per volume. All right. Now a couple of key features about weight per volume is that oftentimes in a clinical setting when we're giving fluids, like in the picture I have normal saline there. If I'm giving a fluid, you know, it's often written as a percentage. And mass per volume or weight per volume is typically how that's given. So let's kind of talk about, you know, how we work with this. So first off, if we're going to work with a milligrams per deciliter, anytime you see milligrams per deciliter, I always do this. I always think instead of milligrams per deciliter, I think milligrams per 100 ml. 1 deciliter equals 100 ml. You guys can work on the conversion later. All right, but understand that 1 deciliter equals 100 ml. So I just converted it to something easier for me mentally to think about. Okay, so this is milligrams per 100 ml. It's a ratio, right? So now I can say, well, this is, you know, milligrams or grams, you know, depending, per 100 ml of solution. Well, I can work with 100 for percentage reasons. Right? It's easy to make to come up with a percentage out of 100. So as an example, I know you guys know how to make percentages out of 100 because when we take an exam, for example, if I gave you 100 questions and I said you got 25 correct out of 100, then you would know that your score was 25%. Right? If I said you got 5 correct out of 100, you know it was 5%. Right? So 100 makes it easier for me to work with percentages. Because we're talking about milligrams really out of 100 mLs. That's really our percentage. So as an example, if I said 1 milligram per 100 ml, and I want a percentage of that, I would say, well, that's 1%. Okay, that's like a 1% solution. 1 milligram out of 100 ml is a 1%. 10 milligrams out of 100 ml is 10%. It's easy, right? Now, let's give you a sort of a real-world example here. Over here, I said 5% glucose solution. Now, this is real-world because actually solutions that you can get are sometimes referred to as D5W, which stands for dextrose, which is just D-glucose, 5% in water. That's what D5W stands for. So that's 5% glucose solution. This is a weight by volume, okay? So that oftentimes, and I should make sure I emphasize this, that when you get these fluids that you give patients, it's in grams, not milligrams. Okay, so we would say 5%, so that'd be 5 grams per 100 ml. 5 grams per 100 ml. So the question is, well, you know, how many grams is that in the entire liter? Because oftentimes these bags of D5W come in liters. Well, you can just multiply the top and the bottom by 10, right? Because if I multiply the bottom by 10, that gives me 1,000 milliliters, which is a liter. And I also have to do the same to the numerator. So that would be 50. So you can use the formula I have there. You can just do the quick and dirty math, which I just did. That would come out to about 50 grams per liter. 50 out of 1,000 mils is, again, 5%. Right? Now let's take another example here. Let's take the 0.9%. Now this is normal saline, another commonly used fluid. All right. Now these often come again in liter bags. So let's take 0.9%, and this is sodium chloride. Sodium chloride. So the question is, how many grams of sodium chloride are in that entire liter? Right, well, you know, it's 0.9 grams per 100 ml. 0.9 grams per 100. That's again our 0.9%. Multiply the top and bottom by 10, right, to give me my liter, and I get 9 grams per 1,000 ml or per 1 liter. So there's 9 grams of sodium chloride in that 1 liter bag, right? This is a weight by volume. This is how I kind of make that calculation. Not that you necessarily have to make this particular calculation, but we do need an understanding of it, all right? Because it will come in handy later. Now, the next step now is to think of this not in terms of weight by volume, but in number of molecules per volume, right? Because with weight, right, and you know, when we compare weight towards number of volume, the difference is, you know, is clear, because in weight I could say, you know, I have 10,000 kilograms of cars versus 10,000 kilograms of pencils. 10,000 kilograms of cars might be, you know, a couple cars, but 10,000 kilograms of, say, pencils mean many, many more pencils, right? So we have another way of looking at it, saying, well, what's the number of the actual molecules, right, in that volume? And that's where moles come in. I know you guys have heard of the moles, Avogadro's number, 6.02 times 10 to the 23rd, right? Which really just represents, this number, like what we would say like a dozen of something. Like I have a dozen donuts. I have Avogadro's number of this particular molecule. Okay, so that's our fixed number, 6 times 10 to the 23rd. So let's take a look at the example of that. So this is telling me how many molecules are in the volume. All right, so if we take a look down here, molecules per mole. Again, anytime you have a mole of something, you have 6 times 10 to the 23rd. So if I had 6 times 10 to the 23rd glucose molecules, right? Again, that's like saying I have a dozen of these glucose molecules, all right? 20's not a dozen, it's a lot more. But let's say I have this amount of glucose molecules. How much does that weigh? Oh, it weighs 180 grams. Well, what if I have 6.10 or 6 times 10 to the 23rd sodium chloride molecules? How much does that weigh? Well, it weighs 58.5 grams. Well, what that tells you is that glucose is larger, right? Is larger than sodium chloride. It's the same number of molecules. It's 6.02 times 10 to the 23rd for both of them, but glucose weighs more because glucose is a larger molecule, right? Now, with that being said, why do I care that there's what the number of molecules actually is? Well, this actually affects fluid shift. Number of molecules plays a role in predicting how fluid will behave. It's one of the features. And so I'm going to come back to that. Okay, so the units of concentration here. This is now where I want to talk about the equivalents. Remember, I mentioned on the previous slide, you saw milliequivalents. And I said, well, what is a milliequivalent relative to, say, a millimole? Okay, here's the difference. So if I have sodium, a normal extracellular amount of sodium or concentration would be about 140 millimoles per liter. Millimoles, all right, is the number of sodium molecules in that volume in 1 liter. Okay. Now, again, you notice I said millimoles, so these are very tiny amounts. This has not even reached the total amount of a mole, which is 6.02 times 10 to the 23rd. It's less than that. Okay, so it's 140. Now, the equivalent, you notice that the sodium, the milliequivalents would be 140 milliequivalents per liter. They're the same. So what is really the difference between the two if they're exactly the same? Well, the difference depends on the valence, okay? The valence, to remind you, in these electrolytes here, what you see is sodium has a valence of 1. It has 1 positive charge, right? Another example would be potassium has 1 positive charge. Many of the electrolytes, okay, that we're going to talk about, a lot of the main ones, are actually only have a valence of 1. And in fact, they don't go much higher. We have ones that have a valence of 2, like calcium or magnesium, for example. Most of them are going to have usually either 1 or 2, for the most part, okay? All we do is we multiply the number of moles or millimoles times the valence. It's a very simple calculation. So if I have 1 mole of calcium, all right, calcium is a valence of 2. So it's 1 mole times 2, and that would be 2 equivalents, all right? I could convert that to millimole. I could say, what's 1 millimole of calcium? It would equal 1 millimole times 2, which would be 2 milliequivalents. We just multiply it by its valence. And if I want to go the other way, if I have the equivalents and I want to know what the millimoles are, I just divide it by the valence. I divide it by 2 or I divide it by 1. Okay? So it's easier to say that anything with a valence of 1, like sodium, potassium, hydrogen, they're all going to have, their millimoles are going to be the same as their milliequivalents because they have a valence of 1. However, things like calcium and magnesium, you have to remember they have a valence of 2, so you're going to have to multiply or divide by 2. So, last but not least, osmoles. Osmoles are very important because we're going to be talking a lot about osmoles in the next slides to follow, because this is really what's going to dictate a lot of fluid shift. Now, I alluded to that already by talking about molarity. how knowing the number of molecules in the solution, right, is important. What's the difference between an osmol and, say, the molarity, or in some cases the osmolality? We're going to use osmolality and osmolarity interchangeably. The reason I get away with this is because the concentrations we're working with are very, very small, so the difference between osmolality and osmolarity are minimal. Chemists would be very mad with me. So if I take an osmol, what is that? That's just the moles times the particles that they times the particles or the molecules that they are in solution. The number of particles or molecules in solution. So what does that mean? So let's take an example because it's easier. So how many osmoles are in 1 mole of sodium chloride? So again, remember we have sodium chloride. I have 1 mole of sodium chloride. When you take sodium chloride, all right, say salt, table salt, right? If I took that and I put that into water, what happens? It dissolves. When it dissolves, what's really actually happening? The sodium and the chloride, which are bound by an ionic bond, break apart into sodium and chloride atoms, right? So they separate in solution. So what we're saying here is that if I add sodium chloride to water, it breaks up into sodium and chloride, all right? It separates. So what I need to know is understand that it breaks up into 2 particles. We'll call them particles or we'll call them molecules, whatever, okay? So if let's say I had 1 mole of sodium chloride, well, it breaks up into 2 when it goes into solution. So I have to multiply it by 2. In other words, 1 mole of sodium chloride, since I know sodium chloride is going to divide into sodium and into chloride in solution, I multiply this by 2. So I have 2 osmoles. 2 osmoles. So what it's doing is it's taking into account a feature of the molecule, whether or not that molecule is actually going to break apart in solution. Because the more molecules, the more particles we have in solution, the more it's going to affect fluid shift, which is really important. Okay, so it has nothing to do just with the molarity or the concentration. It also has to do with the number of particles it dissolves into or breaks up into in solution. Sodium chloride breaks up into 2. All right, so therefore I multiply it by 2 and get my osmoles. I really want to know that number. So let's take glucose as another example. 1 mole of glucose. So the question is, does glucose break up in solution? No. Okay, it doesn't divide into anything. It stays as its fixed molecule of glucose. So 1 mole of glucose is 1 osmole of glucose. I multiply it by 1 because it stays as 1 molecule. It doesn't break up. All right, and one last example, so hopefully we can hammer this point home, is let's say I have 1 mole. And remember, I'm using 1 mole right now as an example, but we could easily say 1 millimole to make it more relevant physiological concentration. But if I had 1 millimole of, say, calcium chloride. So calcium chloride, again, this is a salt that's actually has 3 components. And in solution, it's going to break up into calcium, and so calcium and 2 chlorides. So it breaks up to basically 3 particles if you want to look at it that way, okay? So I'm going to multiply it by 3. So when I take 1 mole of calcium chloride, put it into solution, I'm going to have 3 osmoles. All right, so that's the idea. The idea is I want to know how many particles are in solution because that number of particles is going to directly affect fluid shift.